Hello, everyone. I'm Larry Forman, the founder and CEO of Cho Plus. We engineer cells to improve productivity. So why is the company called Cho Plus? We started out engineering CHO cells for increased therapeutic protein production. A headline is 13.5 grams per liter of IgG1 in shake flasks, but that's not why we're here today. So though my background is mostly in therapeutic protein production, I learned a lot about viral vectors in the last few years, and this is the tame version. So basically, there's not a lot of differentiation among different means of producing AAV. A lot of different approaches, but basically it's all too expensive in the end and not productive enough to treat more common disorders. And everyone agrees that improvement is needed. So I want to spend a few moments on this slide. So some of the problems are that these $2 and $3 million treatments for rare disorders, while it's a noble effort, this cannot possibly translate to treating larger scale numbers of patients. If you want to think of 1 million patients for an aggregate handful or 2 handfuls of genetic diseases or genetic treatments, that could come to $2 trillion, and it's not going to happen. Well, the other problem is that even if the cost of production was free, the current state of the art is that you can't produce enough viral vectors to treat hundreds of thousands, let alone millions of patients. So here's the rub. So the global therapy, cell and gene therapy market estimate up there, it's a wild card, anyone's guess. Basically, it bounces around with effects of licensing of new therapeutics, and then the uptake afterwards. So people have been surprised that after several therapies for rare diseases have been approved, there's very little uptake, and cost has to be a big part of that. So a lot of the shine has come off the gene therapy market, and certainly investment has gone down. And the reason is that people see the problems that I mentioned just before. So if it was possible to produce more doses more cheaply, then there would probably be more uptake as well. So the obvious question is, can we engineer HEC293 cells the way we engineer CHO cells and improve AAV production? And of course, the answer is yes. So this is a list of the problems that everyone here is familiar with, and all except for the last one, immunogenicity, are all related. Low productivity, low full to empty ratio, high cost of production, and the number of patients you ultimately want to treat. Immunogenicity, well, that's related to the capsid, whether it's a custom capsid or standard capsid, and that's something we can't really do much about. So the way we engineer cells, whether it's CHO cells for protein or HEC cells for AAV production, we start out by fusing cells over and over again. And what we're doing is we're creating a library of cells that have shuffled genomes and randomly amplified whole chromosomes, maybe hundreds of thousands of different karyotypes. But then what we can do is we could screen or select this library of engineered cells for phenotypes that are advantageous. Going to the bottom first, for generating cells that can produce high amounts of therapeutic proteins, we look for cells that have anywhere from 10 to 40 times more endoplasmic reticulum per cell, and that's for CHO cells protein production. Well, that's not a factor for AAV production. For AAV, we look for cells that have high amounts of mitochondria per cell, high reactive oxygen species, which is counterproductive. You would expect lower reactive oxygen species to be better, and a high membrane potential. And within that smaller haystack of cells, we can find cells that are very highly productive. So, clicker's not working. Okay, so we started working with AAV serotypes 1, 2, 5, and 9. Together they make up about 2/3 of all the AAV therapies that are in the clinic. And this initial work used the standard triple transient transfection, with your transgene on one plasmid, rep and cap on another, and the helper on another. Sorry about that. Clicker's not working. Okay. Okay. So what we see here is, and I want to tell you first about these groups of bars. So the first bar represents the parent, the second represents a pool of engineered cells, and the third represents screened clones from those pools that are much more highly productive. So when you look at AAV2, for example, we see about a 20-fold increase in specific productivity, and we're up to about 4 times 10 to the 16 viral genomes per liter. So the other problem that people see is that when they do manage to achieve somewhat higher productivity, they see a decrease in the amount of full capsids compared to empty capsids. And what we see here is that we've been able to maintain the percentage of full capsids. So we've got higher productivity, up to 20-fold higher, and we've got similar full to empty ratio. So now we ask the question, can we engineer cells for higher percent full? And here you see the same groups of 3 bars for AAV 1, 2, 5, and 9. And we can get to about double the percent full before purification. So this is a big development. So what else can we do? We started to look at packaging cell lines as a lot of people would acknowledge is very important for going forward in the field to get away from triple transient transfection, or eventually any transient transfection. So basically we have, we've basically stably transfected the cells with rep, cap, and helper, and we only have a single transient transfection with our GOI, or payload. I want to point out here that we have pairs of bars instead of sets of 3 bars because we haven't had the time to clone these out. So these very good results are for engineered pools of cells, which are an average, and since we have very diverse pools, if we were to clone these out, we're confident that we'd have even better results. So we see about a 4-fold increase in titer for capsid production for our packaging cell lines, and we see about a 2-fold increase in percent full. So what we're doing is we're putting together all the nuts and bolts as we go towards fully integrated and stable inducible producer cell lines. We're getting there. So this is just a bit of a summary. So so basically we've got higher productivity, higher percent full for both triple transient and for packaging cell lines. And I just want to summarize some of the new results and discuss a little bit. If anyone is interested in details, see me at a break or we can chat after the conference. So as I said, we're developing more nuts and bolts, more individual procedures that can help with unengineered cells as well as engineered cells, and we're happy to share some of that information, but the fact is that it works much better with our engineered cells than in the unengineered. So for example, when we separate rep and cap, most people use rep and cap on a single plasmid, so when we put the cap gene stably transfected and inducible into the host cells, and then we have cap and helper on another plasmid that's transiently transfected, as well as the GOI. By separating these, we can get much higher productivity. Another double transient transfection system uses a stably transfected helper that is constitutively being produced at a low level, so it's not even under inducible control. And then we have 2 more plasmids, one for rep and cap and one for GOI, and those are transient, and we get much higher production there. A lot of people have asked about stability of our elements that we stably transfect, and we've shown 60-generation stability for stably transfected helper in our engineered cells. We use Sleeping Beauty transposase to do this, and Sleeping Beauty is very good at, though it's semi-random, not quite random, not quite directed either, it does seem to find safe harbors where there's stability for the integrated elements. So we've also been looking at along with applied stem cell and their target system, we've been looking at landing pads to put eventually our GOI into safe harbor H11 loci. And so we would take packaging cell lines that have all the other elements in them, whether under inducible control or constitutive. And with landing pads, we could go from a packaging cell line to a fully integrated and inducible producer cell line in about 4 weeks. We're still working on this, but that's the goal, so that people could go into the clinic with their ultimate producer cell line, not triple transient and doing a changeover between phase 2 and phase 3. Whoop. And last, we've done some two-step purifications. I showed the results earlier for AAV5 production where we had 55% full, and we showed a significant increase in yield, in recovered yield after purification, and ultimately with a higher percent full as well. And that's important to note, because here I want to just show that on the left you see the current state of affairs, where you start out with about 80% empty capsids and 20% full. After purification, you'd like to have about 80% full and 20% empty, but along the way, because you don't get baseline resolution, you lose a lot of your full plasmids. So we're up to 55% full before purification. We intend to get to 80% full or better if we can. And after purification, we hope to get to 95% full, which would be better for patients, lower doses, and probably set a higher standard for the industry. So that's our goal, and you wouldn't lose very much of your full plasmids if you could start out with much higher percent full. Last month, we were awarded our first US patent for AAV production using engineered hex cells. We have numerous patents for protein and hex cells already, so this is new for us. And there are many other patents pending at this point. So looking at this slide that I showed earlier, I think you could see that we're on our way to addressing almost all of these except for immunogenicity, which we really have no control over. So this applies to both CHO cells and hex cells that we work with. We work only with suspension cells. We don't use foreign genes or viruses in the engineering process. We expect fairly routine regulatory approval. And everything we do is suspension adapted. We use commercially available medium that's animal and serum free. And our scale of operation has been shake flasks, but we don't foresee problems in scaling up. That hasn't been our experience in past lives with protein. So what's next? We're working towards our fully integrated and stable inducible producer cell lines. Every time we put some effort into this, and you'll see what I mean, we get higher productivity and higher percent full. We're improving, constantly improving our work with landing pads, and we hope to get that fully integrated in the near future as well. I told you about our aim of 95% full, setting a new standard for purified product. And we're also thinking along the lines of replacing HEC-293 with another human engineered cell line. And before the change of US administration, we had some positive words from Peter Marks, who's no longer there, that if it's safe and if it meets an unmet patient need, then the FDA wouldn't stand in the way, and well, all bets are off now. But we think that if it helps patients, it'll probably be a good thing and be accepted. Cho Plus has a talented and experienced senior leadership team, and while we have While we have 4 lab scientists, all of the work that I showed you today on AAV was performed by Kathy No, our very talented Associate Director of Cell Engineering. So thanks a lot for coming by, and I'll see you at the break or anytime you wish. Thanks.